I'm going to give you a set of questions to look for while you're reading that chapter. It's not that you're necessarily going to hand in those questions. We're just going to use those as springboards for discussion. There's a couple of purposes for that. One is to make sure you understand what you read. And the other one is to take what you read and try to apply it to something that's in your life or in the world, right? Because it's not important enough just to read and understand. It's also about how do I apply that to my everyday experience. Because if I can apply it to my everyday experience, I'm going to retain it much better, right? And that's going to be true when you get into science courses. Okay, so this is what we're going to do. We're going to sort of read the book, comprehend it, try to apply it to something in our lives. The book is going to tell you in sort of a layman's language, recombinant DNA, what happened, how they came up with it. I'm going to give you a little more detail because you're going to need that detail when we start doing the gene cloning experiments. Okay, so I'm going to keep it simple, but there are certain basic principles that I want you to understand about DNA, RNA, and proteins that's going to make it easy to understand when you do the experiments. This is very informal. If you have a question, I'm going too fast, you don't understand, stop me, raise your hand, ask a question, right? Okay, it's okay. We can go through this in a very slow pace, right? But it's important that you get this material so that we can then, when we start talking about all the other stuff, you're able to understand it. So what is DNA? DNA is just an abbreviation for deoxyribonucleic acid, which is a biological molecule. But what is it really for us? It is the genetic blueprint. It determines what you look like. So every cell in your body has DNA in it. And this DNA is information. It's like a cookbook inside the cell that has all the information that your body needs to make you who you are. It turns out the information only has 4 letters. It's not like the English language where there are 26 letters. There's only 4 letters in the information. The letters are A, G, C, and T. And they stand for particular beads on a string. And all DNA is it's a long strand of beads that have these letters in different orders. So, I've got the information. I told you, the DNA is just the information, right? It isn't the structure. It's the information to make it. It's the cookbook. The question is, how does the cell take the information and make the structure or make the function? Because that's the second key. It's not enough just to have the information, but you got to be able to take that information, read it, and make a structure or a function that makes the organism. Because the organism is the structure. It is the interaction of cells in the body. And this all has been characterized into something called the central dogma of biology. And anytime anybody says central dogma, you know it's important, right? This is one of the explaining theories of biology, of how inheritance is explained and how we take that information and we make it into structures and functions. And it's a very simple thing. It essentially says the information that's in DNA has to first be transcribed into another molecule. That molecule then is read to make a protein, and that's called translation. So we have an intermediary here called RNA. We can't take the information directly here to make a protein. We have to have this intermediary called RNA. So going from DNA to RNA is called transcription, and then from RNA to protein is called translation.